Good afternoon, everyone. Welcome to the Wednesday afternoon lecture. It's a particular delight for me to be able to do today's introduction because the speaker was a postdoc in my lab a little more than 20 years ago, right at the point that I had just arrived at NIH from the University of Michigan and was setting up various experiments in molecular genetics. And Dr. Wijmenga, Siska, as I usually refer to her, joined us along with a couple other people who are here, Paul Liu, also at the same time, Mike Erdos in my lab all that time, and now including. And we did some pretty interesting things at that point, particularly in the area of the molecular biology of the M4 type of acute leukemia. But Siska has gone on to do amazing things, and now as a department chair in the Netherlands, comes to speak to us today about germs, genes, and host defense. She got her PhD in Leiden, working on facioscapulohumeral muscular dystrophy, FSH dystrophy, which was at the time quite a puzzle, and ultimately the puzzle got figured out. And then after postdocing in my lab here at NIH, which was a wonderful experience for me and I hope for her, she then moved to Utrecht, where she was assistant professor and then full professor. And then since 2007 has been at University Medical Center Groningen in the Netherlands, where she is now head of the genetics department. She also has an adjunct professorship in medicine at the University of Oslo. She's received a number of awards, including being elected member of the Royal Holland Society of Sciences and Humanities. She received last year the Spinoza Award, which many people consider to be the Dutch Nobel Prize. And she's a member of the Dutch Royal Academy of Arts and Sciences. Her research, which she will tell you about, has been devoted for quite a few years on understanding the molecular basis of the host immune response and how genetic variation leads to dysregulation of that proper response, and particularly how that plays out in celiac sprue and in systemic candidiasis. And to study that, she has founded a number of cohort studies, which I suspect will appear in her presentation, digging deeply into trying to understand in human biology how can we learn about the immune system and its role, and particularly how can we think of the human organism as a superorganism, including the microbiome, which obviously has gotten a lot of attention lately, and in fact, Siska's lab has contributed very substantially to what we're beginning to learn about how the microbiome intersects with human cells and human organism to create health or disease. So please join me in welcoming Professor Siska Wijmenga.
Well, thank you, Francis, for this lovely introduction. It has been a great pleasure to spend here the last 1.5 days. It was really fantastic to come back after indeed more than 20 years. And as you can see from this slide, you may recognize the people over here, Francis and Paul. We all looked a little bit younger right then, but I think it's quite special that even after such a long time, we still kept in close contact. But really being a postdoc here was also an important turning point for my scientific career, because it was here where I learned that we could also really work on the genetics of common complex diseases, really inspired by the work that Francis started off on type 2 diabetes. And for me, that was really an eye-opener, knowing that more than 50% of the world will suffer from chronic diseases, and all of us, as we're sitting here, probably will be gaining one of those diseases over our lifetime. Also, that would be fantastic if we have a somewhat better understanding of those diseases and eventually can even prevent or treat them. And that really led to the main research interest that I started off when I started my own lab in Groningen on celiac disease, and that has been the main theme since then, but I won't talk about it at all today. So where is Groningen? So this is the Netherlands, which is a tiny country squeezed in between Germany and Belgium, and close to the UK. You probably all heard of Amsterdam, and Groningen is really up in the north over here. And maybe you have been heard about that recently because the Nobel Prize in Chemistry was awarded to my colleague in chemistry, Ben Veringa, and we were all pretty proud of Ben, as you can imagine. But Groningen also has a large university. It was established more than 400 years ago, and it has more than 30,000 students, and it's one of the largest universities in the country. And associated to that is also a large medical center, and that's really where my department is located. And that medical center has about 1,400 beds, and it's the largest teaching hospital in the country. Okay, well, coming back to those common complex diseases, you're all aware of the fact, of course, that over the past 10 years or so, people have been doing those genome-wide association studies. And I can hardly believe of any trait or disease that has not been used in those type of studies. So we have now loci for almost all of them. And the big, and the same happened, of course, for celiac disease, where I have been devoting quite a lot of my scientific career to. So for celiac disease, we have now 43 different loci that explain about 50% of the heritability. And I think one of the most intriguing observations that we made is that the loci that we identify for celiac disease are partly shared with other autoimmune diseases like type 1 diabetes, rheumatoid arthritis, Crohn's disease, and so on. And that really means that if we're going to discover a little bit more about the biology of celiac disease, at the same time we're also learning about those other diseases. But this post-GWAS era is really not an easy route to take. So it probably will be a couple of more years before we really make another major discovery in trying to understand how all this genetic variation really contributes to disease biology. And that is really what we have to do now. We have to understand how all those genetic variants really translate to phenotypes, either being disease phenotypes or more general phenotypes in the general population. And this is a pretty black box. It is clear, however, that we have to go beyond the genome. It's not enough to only look at genetic information. We have to do a more holistic approach. And for that is important usually to also have information on, let's say, the transcriptome, the metabolome, proteome, epigenome, exposome. There's lots of exposures we are confronted with all the time, and also the microbiome. So both in this country as well as in the Netherlands, there is a lot of excitement that people think that we really will benefit from studying large cohorts of people from the general population. And that is for many different reasons. One of it is it's usually large sample sizes. We collect many phenotypes from those people, including omics data. What I think is extremely important is that the data is collected in a standardized way, and that we follow people prospectively. And that means that we have data collected before they get a particular disease, during the time of disease, and also after disease onset, and hopefully at the time they're being treated if possible. So you have this wonderful project here in the US, the Precision Medicine Initiative. And we're trying to set up something very similar in the Netherlands, which is called the Health Research Infrastructure, or HealthRI. And really one of the cornerstones of HealthRI is our biobanking initiative that has been ongoing now for approximately 10 years. And that is really because we have many biobanks traditionally in the Netherlands, and they are both population-based as well as clinical cohorts. And as of today, there's more than 200 different ones, and they're all united in this biobanking research infrastructure that really tries to harmonize the data and also make it possible that people can connect all the data from the different single initiatives. And those biobanks are really spread across the country. So they really come into many different flavors. We have smaller ones and also larger ones. They are partly healthy population and partly specific phenotypes, disease phenotypes, and they're both cross-sectional and longitudinal. However, if you take all the people that participate in any of those biobanks together, it turns out that about 6% of the Dutch population, which is some 1 million inhabitants, are already participating in biobanks. And that's really a fantastic resource that we can use to at least understand a little bit better what the effect is of genetic variation in the general population. So to give you a couple of examples of biobanks that are commonly used and you may have come across by reading papers on, let's say, GWAS studies. We have a big one in Amsterdam, which is the Dutch Twin Registry. It has about 200,000 people participating. And that is, of course, very important to also establish what is the heritability of traits we are studying. We have the Leiden longevity study that's really focusing on people that become extremely old. We have the Rotterdam study that follows people over 55 years in age from Rotterdam. And it's smaller, but it has follow-up data for more than 25 years, so also a lot of information on natural history of diseases. And then in Groningen, we have the LifeLines cohort study that was started some 10 years ago, and that has 167,000 people participating, and this is the largest population biobank in the Netherlands. So a lot of the work that we do today in my group is really centered around the LifeLines population. And really this BBMRI initiative has been extremely important for the research community in the Netherlands and was, for example, instrumental in a project that I led a couple of years ago, the Genome of the Netherlands. And that was really meant to get a better understanding of the genetic variation in the Dutch population. And what I think is really striking, the Netherlands is a tiny, tiny country. It's only 100 miles wide, and if you go north-south, it's only 175 miles. Yet, if you look at genetic variation, we see major differences. So people in the north are completely different from people in the south. And that is important because if you start to use clinical sequencing and you want to infer whether a variant that you find has anything to do with your phenotype, but when your patient is not matched very well with your controls, you really may run into deep trouble. So this resource that we now have for a couple of years is really used as a major reference for clinical sequencing in every clinical genetic center in the Netherlands. Okay, but what I will do for the remainder of my talk is give you 3 vignettes of recent work that is coming out of the Dutch biobanks. One is about factors that we identified that affect gut microbiome composition. One has to do with variation of cytokine production. And the third one is factors that are involved in host response to fungi. And much of this work is really centered around 2 relatively small cohorts. One is a subset of the bigger LifeLines cohort, which we call LifeLines-deep. And the other one is a cohort, and that's 1,500 participants. And the other one is a cohort of only 500 individuals from the area of Nijmegen. And this is really a project that we run together with Ramnik Xavier from the Broad Institute and with Mihai Netia from the Radboud University in Nijmegen, and that we collectively call the Human Functional Genomics Project. And there's even actually more smaller cohorts in this project. So what about gut microbiome composition? Well, the first question you may ask yourself is, well, why would you study gut flora at all? And for me, this paper that was published a couple of years ago in Gastroenterology was really an eye-opener, where they showed that if you transplant fecal material from healthy lean donors to people that were obese and suffering from metabolic syndrome, by just doing this simple single intervention, the metabolic syndrome really improved in a period of 6 weeks' time. So really telling us that the gut flora may be a target to modulate human health and might have dramatic consequences for chronic diseases, for example. Well, our gut microbiome is really essential for human physiology. On the one hand, it has a very important role for our metabolism. It synthesizes, for example, certain vitamins, but is also there to digest very complex molecules like starch. And on the other hand, it's very important for our immune system. It really trains our immune system and is also a barrier against all kinds of pathogens. And if we look a little bit more in detail, then we see that really the majority of the bacteria, of our bacteria, can be found in our gut. And in our gut there is is somewhere between 500 and 1,000 different bacterial species. And if you think about it, it means there's 150 times more genes produced by our bacteria than by the host genome. But given that there are so many different species in our gut, it also means that all of us have a different gut microbiome that can vary both in the composition as well as in the diversity. So we can have all different numbers of the same microbe. And until recently, it was really hard to get a good understanding of the bacteria that are there, because it has been really difficult to culture bacteria from your gut, because we really don't understand the culturing conditions. But now with next generation sequencing, we can of course do that. And there's really two ways that people try to approach this. One is by sequencing the 16S rRNA gene, and then you get an insight into the taxonomy, pretty broad. But you can also sequence everything, so shotgun metagenomic sequencing, and then you get much more information at the species level. You not only sequence bacteria, you also sequence everything else that's there, like fungi, parasites, and there's lots of viruses actually. But since you sequence every gene of your bacteria, you kind of also can infer what is the functional capacity of the bacteria in your intestine. So you can move a little bit closer to what they are actually doing instead of what are the bacteria that are there. And that is very important if we really want to understand how the gut microbiome really influences our physiology. But really thinking about the gut microbiome and reading papers when we started this project about 5 years ago, it was really clear that it was nobody really knew what is a normal microbiome and what are really the factors that influence that, either being genetic, phenotypic, or environmental. And since we have this LifeLines-deep cohort with lots of data on individuals, we thought, well, this might be an opportunity to really study that. So for all of the 167,000 people participating in LifeLines, we have about 1,500 phenotypes. And that is data on diet people take, medication, smoking habits, but also information on all kinds of physiological parameters like blood pressure, blood lipids, and so on. But then in addition, in this smaller cohort of 1,500 individuals, we have very deep omics data. So we have genotypes, we also have methylation data, transcriptomics, proteins from blood, so cytokines, adiponectins. We measure metabolites, both in exhaled air, which are the volatile organic compounds, as well as in plasma, and we have both 16S rRNA sequencing as well as metagenomic shotgun sequencing of our gut microbiome. So that is all based on stool samples. So we set out a comprehensive study to really see, well, what is the effect of those factors on the gut microbiome. So not all the participants did participate in collecting stool samples. We had stool from just over 1,100 people. And by sequencing them, we could distinguish more than 600 different bacterial species. And then we correlated that with 78 different factors on diet people were taking, 44 categories of drugs, 5 categories of smoking, 39 different diseases that were present in that group of people, and 41 intrinsic factors. And then you should think about factors like age or gender, lipid levels, etc. And interestingly, what we found, the single most important factor that really determines variation in our gut flora is our stool type. So how does your stool look like? And we were measuring that from people, giving them this, what we call the Bristol stool chart. And I think it's very self-exploratory. The only thing you have to say is a type 1 till 7. Being a very hard stool, type 1, to a very diarrhea form if you have the type 7, and everything in between. But it turns out it really matters with respect to how diverse your microbiome is. And that's depicted in a picture that came not from our publication, but from a group in Belgium that did more or less the same, is that if you have a diarrheic form of stool, then you probably flush out a lot of the bacteria in your intestine. And all of a sudden you have a much lower diversity of bacteria compared to people that have the more harder stool types. And in general, it is considered that more variety is better. So that means that you rather want to be in this corner and not so much on the diarrhea side. But if you would not know this information, it also means that this can be a major confounder of your data. And so far, I haven't seen that all the microbiome studies are collecting this type of information. But given that it's such a major determinant, this is really something people should take into consideration and really control for. Well, not surprising, of course, is that what we eat also very much determines what's happening with the bacteria in our diet, because they're also in part dependent on our food. And again, we have two categories. We have food items that really enriched our diversity, and those are the ones in green, and again, more diverse is considered to be better. And there are food items that are not as good because they reduce the diversity. And it's interesting to see that the items that really increase our diversity are also including coffee and red wine, and that's maybe not something you would expect up front, but also fruits, vegetables, and yogurt, whereas beers and soda is something you probably should avoid. Well, since we also had medication use from the individuals, we could also look at what is the effect of taking medication on your gut flora. And again, we saw that a lot of the medicines people take have quite some effect on your gut microbiome. And by doing so, that can, of course, have all kinds of secondary consequences like enteric infections, gut inflammation, or even have an effect on host metabolism. And if you go through the list, there is lots of medications that are quite widely prescribed, like statins, oral contraceptives, but also antidepressant medication. But really what is on the top of this list are the proton pump inhibitors, and they really have a major effect on the variation of our gut microbiome. And this is a very widely prescribed drug. We also know that if you continuously use proton pump inhibitors, you really increase your risk for C. diff, and there's a lot of epidemiological studies that really show that, and we also know that there is really an increase of people that are hospitalized with C. diff. So when we looked into the gut microbiome of PPI users, it was really clear that we had major shifts in about 20% of the bacterial taxa. So we see that people increase all the bacteria that are given here in red, so they have more Enterococcus, Streptococcus, Staphylococcus, and Escherichia coli. And these are potentially kind of the bad bugs in your gut. And they really reduce the more beneficial taxa, in particular the bifidobacteria and the ruminococcus. We also know that if you take PPI, you really reduce the pH in your gut, and that probably is the reason why you get such a big change in the local environment. That on the one hand you allow other bacteria to take over niches that are emptied, and also pathogenic bacteria that normally would not survive because of the acidity, all of a sudden can survive. What was interesting, however, that when we also had microbiome from the oral cavity from a small subset of the people, and we saw that the bacteria that were in the gut from the people with the PPI was much more reminiscent of the bacteria in the oral cavity, really telling us that you get a shift from your bacterial composition. So I think we have to be a little bit more careful in prescribing PPIs because of those dramatic effects on your gut microbiome. So if we put all the data together that we analyzed, and I only showed you a couple of examples, then we had in total 126 factors, and again, mainly coming from medication, diet, and some of the intrinsic factors like lipid levels. They explain nearly 20% of the variation in our gut microbiome, and that is a substantial proportion. From twin studies, however, we also know that there are certain bacteria that are quite heritable, and that would suggest that they are not so much influenced by our environmental factors like diet and medication, but maybe more by our host genome. And this is work done by the group of Tim Spector on the twin UK cohort. And it really shows that some of the bacteria are heritable, whereas others are not. So there really seems to be a divide in our bacterial community. So we were really asking ourselves the question, well, can we also detect really an effect of host genetics on the gut microbiome, especially given that there is so much noise from all the other factors coming from the environmental factors. So to address that question, we again made use of the LifeLines-deep cohort. We took out all the people that are on antibiotics. And we had a replication in the 500 FG cohort from Nijmegen, as well as a smaller cohort from Maastricht, and we used those two for replication. So doing that experiment, we saw indeed that there is an effect of genetic factors on our gut microbiome, although this is a pretty modest effect. And we had both association to single taxa, to single bacteria, and some of them were also the bacteria that have a reasonable heritability, so that is also what you would expect, because they're the more heritable bugs. And we also had associations to metabolic pathways. I'll give you a couple of examples of data that we identified, one on the LCT story and one on the C-leptin genes. But this association was also interesting with the genus Blaudia, because we saw a SNP that is really in or near the LINGO2 gene, and that is associated with obesity and BMI. And we know that there is really a strong effect of the microbiome on body weight. So we saw, in fact, many different genetic variation in many different genes that make up the leptin receptors. So we have different leptin receptors, and there's many different genes involved, but all the ones in red are the ones that we identified in this study as being associated with the gut microbiome. So we know that those receptors recognize intestinal microbiota, and after activation can really modulate the production of pro-inflammatory cytokines. So really providing the link between the microbiome and the immune system, probably modulated in part through our host genome. Well, another interesting observation we made was with individuals that carry the genotype that makes them lactose deficient. So you probably all know the story that we need the lactase enzyme to digest dairy products, because that can break down the lactose, which is the sugar in dairy products. And the G genotype in the lactase gene is really the genotype that's associated with being lactose deficient. And people that carry that genotype have much higher levels of bifidobacterium than people that don't. So these are the carriers, and these are the people that only have one allele or are completely wild type. And you see there's a major difference in the amount of bifidobacterium in the gut of the GG carriers. Interesting, however, this is only the case when people drink a lot of milk. And this sounds a little bit weird, because if you're lactose deficient, you would think that those people do not consume milk because they cannot digest it. Well, that does not be the case. So since we have this nice cohort, we can also check how many people have this LCT genotype and how many of them still drink milk. And that's quite a reasonable amount. But having this G genotype and drinking milk, only then you have increased levels of bifidobacterium, really telling us that if you drink milk but you have the bad genotype, you kind of compensate for that by having increased levels of bifidobacteria, because this bacterium can also metabolize lactose and kind of diminish the effect of not having the gene yourself. So I think this is a very nice example of this gene by environment interaction, and there's probably many more to be discovered in the data that we have generated. But these will be pretty hard, of course, to detect as well. So far we have mainly been looking at whole communities, but in order to really understand how bacteria provide their function, we really have to move on to single strains and eventually also understand what are the metabolites that are being made by the bacteria and how do they signal back to the host. So to do that, we have collected a much larger sample size of 10,000 people, but also in such a way that we still can culture single bacteria, and that will really help us hopefully to go a little bit more deeper into more functional studies. At the same time, we're also doing longitudinal experiments because we really have to understand what is cause and consequence, because everything I showed you so far is just association. And that also includes 1,500 newborns, where we can really also see how this microbiome is kind of teaching our immune system as well. And then we have to also enrich the data. So we did also fecal metabolites, for example, to really understand a little bit better what the bacteria are all making themselves. So that was my first vignette. So now I want to move on to the second vignette, and that is about factors that influence our variation in cytokine production. And this comes back to, of course, my interest in autoimmune diseases, because what is really going on in autoimmunity is that your immune system is not working properly. It's overreacting to something it should not do. But on the other side of the coin, you also have, of course, an immune system that is not working good enough and really leads to susceptibility to infection. So it is really important that we have a proper balance to have a appropriate immune response that doesn't go in either direction. And to really, and we are confronted as human beings all the time with all kinds of pathogens, of course, and to really eliminate those, we need both specialized cells as well as a specific set of cytokines that take care of that. But very similar to our study in the microbiome, our current understanding of the variation in the host with respect to the immune system is really limited. So we really don't know what is the normal variation and what are the factors that are really influencing that, either being genetic or intrinsic or exogenous factors. So for that we turned to the 500 FG cohort, which is very similar in design as the LifeLines-deep that I showed you, except that it has in addition a very deep immune phenotyping. So we have a lot of data on platelet activation, we have different immunoglobulins, we have very detailed information of the different cell types that are present. We flow sort 73 different ones. And then we have a lot of information on the production of cytokines in response to different stimuli. And we look to 6 different cytokines and 18 different stimuli. And that all in 500 people. So just some general characteristics of 500 FG. We have a wide age range, but you can see there's really a skewing towards people in the age range between 20 and 30. And that is because these are mainly students being contributing to the study. We have a pretty normal BMI. We have almost equal amounts of males and females. Half of the females use oral contraceptives. About 13% of the cohort is smokers. And then we also have information about the seasonality, because those samples were collected over a 1.5 year time period. So between July 2013 and January 2015. So we can really also see what is the effect of season. And you can imagine that it really matters whether it's winter or summer. If you're, for example, exposed to flu virus, your immune system may be in a different state than when you don't have this exposure. So what we did is we took blood and PBMCs from all those individuals and then stimulated them with 3 different fungi, 7 bacteria, 1 virus, 4 bacterial ligands, and 3 metabolic stimuli. And this was all ex vivo. And then measured after 24 hours TNF-alpha, IL-6, and IL-1 beta, and these can be regarded as monocyte-derived cytokines. And then after 2 days and 7 days, we also measured interferon gamma, IL-17, and IL-22, which we then can regard as T-cell-derived cytokines. So we had for every individual, and we had a little bit over 500, 95 different cytokine measures. And the first question we were asking ourselves is, well, how are those cytokine responses built? Is it so that if you encounter a certain pathogen and you respond with a certain level of TNF-alpha, for example, do you then also have the same level of IL-1 beta and IL-6? Or is it that once you are a high TNF-alpha responder to, let's say, a certain bacteria, are you also a high responder to a virus or to a fungus? So when we looked at the correlation of all the cytokines produced after stimulation with those different stimuli and did an unsupervised clustering, it immediately became clear that all these stimulations in response to bacteria were grouping together, as well as the fungi, as well as the viruses, really telling us that the things are built in a pathogen-specific way. And this is just a blow-up to show it in a little bit more detail. So if you stimulate with LPS or candida or with Staph aureus, in all cases you have a very correlated response with the 3 different cytokines. So really telling us that it's really the pathway that is, the pathogen that is determining this. So that means that if you respond well to a bacteria, you may not respond very well to a fungus, for example. So there is a lot of plasticity in the response to pathogens. But since we also had, of course, the genetic information combined with the fact that there was a lot of variation between individuals, we could also assess, well, is this production of cytokines, is this under genetic control? So if you intersect that 2 types of data, you can just see if, depending on the genotype, people have higher or lower levels of cytokines being produced. So we call them cytokine QTLs. So by doing that, we saw that this is indeed the case. From this small cohort of just over 500 individuals, we saw there's 17 loci in the genome where genetic variation really impacts the level of cytokines you produce in response to a certain stimulus. And this is a circular Manhattan plot, so a little bit different from what you're used to. Here we have chromosome 1 and then all the way to chromosome 22. So the different colors just tell you what is the ligand that we use for the stimulation. So the blue is the bacterial, and we see that really the majority of the cytokine QTLs are in response to bacterial ligands. And then we see a few, the pink ones, and those are fungi. There is a very strong CQTL here, and that is the toll-like receptor gene. And the SNP is really a point mutation in the toll-like receptor 1 gene, so probably pointing to the functional variant. Interestingly enough, we see that in particular the production of IL-6 and IL-1 beta is under genetic control. So out of the 17, 10 of them are either for IL-6 or for IL-1 beta. And we see less so for the other cytokines. And the other interesting observation was when we intersected those cytokine data with GWAS data on either autoimmune diseases or infectious diseases, we saw that these cytokines that we regard as monocyte-derived, so IL-6, IL-1 beta, and TNF-alpha, mainly overlap with SNPs that are also associated with infectious diseases, whereas the T-cell-derived cytokines mainly overlap with the autoimmune diseases, which are also regarded mainly as T-cell diseases. So that is an interesting observation, because on the one hand it really puts function to GWAS SNPs, But it also tells us a little bit about effector cells in those different phenotypes. So being the infectious diseases more monocyte related, and that fits with the fact that these are more innate phenotypes, whereas the autoimmune diseases are more of the T cell type, which is more of the adaptive immune response. Well, since we have so much information on all those individuals, we can also see, well, what is the contribution of other factors on cytokine production? And really the majority of the effect is coming from genetics, so SNPs, as well as gene expression, although gene expression is under genetic control, so they're not independent of each other. And then we see a little bit of effect of cell composition, but not as much as we probably would have expected. We see a little bit of effect of seasonality, but really not so much about age and gender. We probably would have expected a little bit more there, but for the gender, for the age, it could also be that although we have a wide age range, the majority of the people are still between the 20 and the 30 years of age. But what was interesting, that we see also some effect of microbiome on the production of cytokines. So what we're doing now is really, I thought I had another slide, okay. Is to see, can we put all this data together to kind of predict how people would respond to a certain pathogen. And this may be interesting in the future if we want to understand a little bit better who is at higher risk for either getting infectious diseases in, let's say, in hospital people or in the elderly. And it really depends on the type of pathogen with the cytokine, if we can do so. So here's an example. Here we put all the different categories of data that we have together. And if you stimulate with E. coli, we can predict with 80% accuracy how you would respond with your TNF-alpha levels. Whereas if you stimulate with influenza, we cannot do that as good. It's only about 50%. But this is still very preliminary work, so hopefully when we add more information, we can really do this a little bit better, and I think that is quite interesting. So coming back to this microbiome, that explains some 10% of the variation in cytokine production. It's interesting also here that we see some specificity in both the stimulus as well as in the cytokines that we see. And we mainly see the TNF-alpha and the interferon gamma cytokines being affected by bacteria in your gut. And we see a lot in response to candida, for example. So really telling us that if we talk about the gut microbiome, in my first part of my talk, I showed you that medication and diet and genetics has an effect. But now we show that also cytokine production can be under the control of the gut microbiome, in particular interferon gamma and TNF-alpha. Whereas for other cytokines like IL-6 and IL-1 beta, I just showed you that they are much more under genetic control. So we're starting to teasing this apart. But I think this is interesting because it also tells you that maybe through diet or medication, we can at least maybe modulate some of the cytokine responses in future. Well, this is of course very early work, but at least it shows some potential. Okay, so I've only a few slides left. Tell you a little bit about, well, can we now use that information that we generated from the population biobanks to also understand a little bit better infectious diseases? And why would you do that? Well, again, infectious diseases are a major health problem. About 1/3 of the population dies because of infection. And we only have a very limited understanding as of today what are really the genetic factors that control that. So we set out to study candida because this is a very serious bloodstream infection that kills about 150,000 people a year. Well, it has been really hard to do genetics on diseases like systemic candidemia. And that is because it's very, very difficult to get large enough cohorts to study. And over the years, we have been studying a cohort of 217 cases. and I think you all start laughing if you're in the GWAS area, because we're really used to study thousands of cases, and we did for celiac disease, for example, 12,000 patients. But this is really the largest cohort that we have today, so we have to deal with that. Well, nevertheless, a few years ago, we did a GWAS study using the immunochip, which is only a limited number of loci in the genome. And much to our surprise, we identified 3 loci with genome-wide significance. And if you look at the allele frequency as well as of the odds ratios of those loci, we see something that's completely different from what we are used to in the other complex diseases. We have rare alleles, and they have a pretty high odds ratio, so they have a much stronger effect than what we normally see. Normally it's in the range of 1.2, 1.4. So already telling us that the genetic architecture of at least this infectious disease is quite different from an autoimmune phenotype. Nevertheless, the loci we identify are also identified for autoimmunity. So it's interesting now to see if they're really going into opposite directions, and we're doing some work on that at the moment. But you also see that in this Manhattan plot, there is a lot of signal that is, of course, below this level of significance. And because of the small cohort we have, it's impossible to boost that. So we were wondering if we can make use of the data that we have generated in our biobanks to see if there's any interesting signal in that as well. And for that, we turn to both LifeLines-deep as well as to 500FG. So for LifeLines-deep, we have transcriptomic data, and that really helps us to do a standard eQTL analysis. So we can identify, if we have associated SNPs, what is really the gene that is under the control of that SNP. Depending on the allele you have, you make higher or lower amounts of that particular gene transcript. So that really links SNPs to genes. On the other hand, we have the 500FG, where we have the stimulations with the different pathogens, and now we can see in the same individual, is a gene responding to a particular pathogen or not? So is it really a candida response gene? So if we would have the situation that we only see a signal when we stimulate with candida, but not with bacteria or viruses, then at least the gene has something to do probably with the infection with candida. So when we did an eQTL analysis, actually from all the SNPs in this green area, this was one of the interesting SNPs we identified on chromosome 7. So the p-value was, of course, not genome-wide significant. It's, again, a rare SNP with a reasonable odds ratio. But if you carry this SNP, you really impact the levels of the SERPIN1 gene. So we didn't have any homozygous carriers, but we had people that were heterozygous. And you see that the level of expression is dramatically lower compared to the people that are homozygous for the wild-type allele. So the risk allele lowers the levels of SERPIN1, and SERPIN1 is really the same as alpha-1 antitrypsin, and this may ring a bell to you. When we went to the candida-induced stimulations, we also saw that this gene was really induced upon stimulation with candida. So here we have no stimulation at all, and then after 4 hours we already see some induction, and after 24 hours we see an even much stronger induction. So again, providing some evidence that this may be a gene that has something to do with candida infections. So then we went to mice that have overexpression of the SERPIN1, because the deletion is I think, is lethal. And when we infected those mice with candida, we saw that if you have high levels of alpha-1 antitrypsin or SERPIN1, you really survive the candida infection, whereas the wild type, they all die after 6 days. So really telling us that this gene is really important for clearing the candida. And remember that the individuals that were having the T allele have really lower levels, and that would mean that they cannot clear the candida as easy as possible. So this raises some interesting questions that maybe by raising the levels of alpha-1 antitrypsin, this may be a future option to maybe treat systemic candidemia. Of course, this needs to be worked out in much more detail, but it really shows us how we hopefully can move on from those observations and how we can really kind of bridge population biobank work with more clinical cohorts. So I gave you 3 examples, and I hope I've showed you that by studying the gut microbiome, we may get some opportunities on how we can really improve our gut health by, for example, changing our diet or changing our medication. I think from the work on the cytokines, I find it exciting to see that maybe in future we can even predict how people can respond to specific pathogens, and this may be important in an aging society. And by combining the data from the population data with a clinical cohort that in itself is underpowered, we may find new inroads into fungal infections. And if you're interested to read more on the Human Functional Genomics Project, just last week we had 3 papers out in Cell that really talk about the effect of host and environmental factors on the microbiome and also on the genetics on cytokine production. Well, with that, I would like to stop, but really thank again Mihai Netia, who is my long collaborator on this Human Functional Genomics Project, and really the 4 very talented people in my group that did all the work I talked about. Yang Li and Vinod Kumar have been doing all the cytokine and candida work, and Jingwang Fu and Sasha Zernakova have been doing all the microbiome work, of course in collaboration with a lot of other people. And thank you for your attention.
Siska, thank you for a really interesting set of experiments and examples. And we have time now for some questions. Please use the microphone so people watching by video can hear you, and we can start right over here.
Just one simple question. Within your cohorts, did you include the parameter of implanted medical devices? The reason I'm asking this is they can predispose to biofilms, and biofilms, of course, imply some type of a bacterial interaction which could interact with the other parameters you are using. I think if you go through the literature, you'll find that biofilms and medical devices, what I'm talking about is chronic indwelling, such as a pacemaker, an orthopedic implant, and now with the insulin pump being probably in Europe being fully approved, this would...
A really good point. I don't think we have that information, although I should go back to the questionnaires because there's so many different questions, of course. But it's definitely something I'm going to check out, yes.
Okay, thank you.
Good point, thank you.
You have identified 2 groups in terms of interactions that falls into the peripheral blood monocyte versus the other group of innate immune response in terms of TNF, interferon versus the interleukins. So in the autoimmune disease like rheumatoid arthritis, how much of the contribution might be coming from the microbiome versus the host immune response? Is there any way you could differentiate? Or at some stage probably they are working together to make the patient sicker than what it should be?
Yeah, so that's also an interesting question. I don't think people have done so much on the microbiome in those disease phenotypes. I know that people are now starting to do it in inflammatory bowel disease, and I think they are really the group that's really the front runners. And I haven't really seen that type of data coming out.
And a short question, you mentioned about the therapy. So how much do you need for the therapy effect? In one of the slides you showed that there are cases where things might work for the benefit of the patients using microbial therapy.
Yeah, so that is of course a really good question, and I think we really need more longitudinal data to also show how quickly is the microbiome changing, right? Because we did a very short intervention with a gluten-free diet in healthy people for 4 weeks, and we didn't see major changes. We see some changes at the strain level, but not really a major difference. And that might not be a strong enough perturbation of your microbiome. And the question is, what happens if you do more dramatic changes? And these are really studies that need to be done. We have to be able to do interventions and really follow people on a day-by-day basis.
Thank you.
Thank you very much. A couple of questions. In the studies that you mentioned with regard to medicine and microbiome, have you noticed whether the vaccines could have a change in the environment and the ratio of benefit to hazard ratio in the microbiome nature changes? That's one question. The other
Can
one,
I address that one first? Otherwise I'll forget all the questions.
Okay.
So we haven't looked at vaccination here, and that would be fantastic because, well, in the Netherlands we have a program of vaccination in kids, and these are of course adults. People get flu vaccination, of course, and that's something which we don't monitor, unfortunately. We're setting up our birth cohort now, and there we specifically make sure that we sample before and after the vaccination periods to address that question. Yes. But that will be a while before we have data on that, so probably another 5 years or so.
Yes, thank you. The other one is, you mentioned that the bacteria usually has the innate immune involvement, and the autoimmune disease have the T cell, the adaptive immunity. Is this because you are comparing the acute inflammatory response versus the unresolved? If the infection continues and you look at it at the later stage, you may see the involvement of the T cells.
Yeah, I think you're absolutely right. And I think this is also what now, at least in the field of autoimmunity, becomes much more clear, that you probably need the innate system first to really kind of start off, and then the adaptive takes over. And yeah, you're right. And here we only look until day 7, right? So we really cannot address that question. But I think the GWAS really tells us that this may in fact be the case.
Thank you very much.
Yeah.
Siska, you showed that interesting circular Manhattan plot with cytokine QTLs. And it was interesting to see how many signals came out of a relatively modest-sized study. So you have clearly big genetic factors here at fault in the expression of these cytokines. But you didn't tell us what those peaks actually represent. Do they lead you obviously towards pathways? Well, for instance, the actual structural gene for what you're measuring, does that turn up there?
No.
Or they don't? Well, that's interesting.
No, that's interesting, yes.
Are there things that do sort of pique your interest there, where the peak is pretty close to something that would make sense as a modifier of expression of those particular cytokines?
Yeah, so first of all, we don't find the cytokine genes themselves. That is an interesting observation. Not many, about half of them by heart are non-coding RNA genes. So really telling us something about gene regulation. Many of them are under positive selection. So this is, well, probably because our immune system. And many of the genes we don't make any sense yet. So we did EQTL mapping, and of course if you go to the paper, there's a list of potential genes, but we really don't know what to make out of that. But it's not the cytokines themselves.
Do these tend to be
genes? So it's probably
genes?
Regulatory.
Do they tend to be genes that are rather tissue specific for the immune system, or are they expressed all over?
That's a good question. That I don't know by heart.
That'd be very interesting to see.
And do they have super enhancers?
Thank you.
Or stretch enhancers.
You mean stretch enhancers, don't you, John?
They are enriched for monocyte enhancers. I don't know if they are in stretch enhancers indeed. Yeah. Yeah, I mean, there's a ton of work that needs to be done. Now we have all this data, absolutely. Yeah.
Well,
so it keeps us busy for the...
Well, people interested in additional questions to pose to our speaker, we're about to adjourn to the library for a reception with some refreshments, so please join us there. But meanwhile, please let's thank Siska again for a wonderful seminar.